It's time to get going, and I'm just thrilled to be here, everyone, on this beautiful afternoon. Nice, cozy afternoon to be in here listening to some amazing science. It is my pleasure to introduce our speaker for today's Rolla E. Dyer lecture, Dr. Jason McLellan, and who I understand this is a welcome back, which is wonderful. Dr. McLellan is a professor in molecular biosciences at the University of Texas at Austin. Before I tell you about his background, I should let you know this stuff. If you're attending this lecture for CME, the code is 57666. If you're watching this online and would like to submit questions, please click on the Send Live Feedback button, and your question will be relayed to the speaker at the end of the lecture. You can submit questions at any time during the lecture. So, now the important things. We are delighted to have Dr. McLellan here today. He is a structural biologist specializing in virology and vaccines. As a postdoc at NIH in the laboratories of Peter Kwong and Barney Graham, from 2008 to 2013, he was responsible for a breakthrough toward developing an RSV vaccine. He determined the structure of the RSV F protein and engineered a pre-fusion stabilized antigen, which elicited very high titers of antibodies in primates and then in humans. The FDA approval of multiple RSV vaccines last year and this year ended a 50-year quest to develop an effective vaccine against respiratory syncytial virus. This has been a great burden to society over the years, especially for vulnerable populations, neonates, especially for whom it has been the leading cause of hospitalization. Building on his pioneering RSV studies, Dr. McLellan's lab also helped determine the first structure of human coronavirus spike protein in 2016, a story we all know quite well. Recently, his group engineered second-generation hybrid-stabilized S proteins that have been incorporated into the design of COVID-19 vaccines that are undergoing clinical trials in developing countries. With 150 publications, multiple patents, and still in his early 40s, Dr. McLellan is regarded as the leading scientist in the field of structure-based vaccine development. Can't wait to see what you do next. Among other honors, he received a National Academy of Sciences Award in Molecular Biology and the UT Austin President's Research Impact Award, both last year, and a AAAS Golden Goose Award in 2020. Those are great. Yeah, those are great. He earned a Bachelor of Science in Chemistry from Wayne State University in Detroit, PhD in Biophysics from Johns Hopkins School of Medicine. So please join me in welcoming Dr. McLellan.
All right. Well, thank you very much for that very generous introduction. I thought today I would talk a little bit about the role that the NIH and NIH funding has played in the development of vaccines for infectious disease, kind of going through and hitting some of the highlights about RSV and coronavirus, and ending on some of our recent work on Crimean Congo hemorrhagic fever virus. So I do have conflicts of interest, which I guess were already shown, but I am an inventor on patents and patent applications and receive royalties related to some of what I will speak about. So for structure-based vaccine antigen design, I really feel like it's one component of this reverse vaccinology approach to vaccine design. And we say reverse because historically you would start with the pathogen and try to isolate the pathogen and weaken it or inactivate it, and then immunize with it and hope that it provided a robust immune response and that you would effectively weaken it so it didn't cause disease. And that worked well for a number of pathogens. And now what we're doing is the reverse is really starting with people who have maybe survived the infections that we're interested in, and they survived it in part because they've mounted a robust immune response. Particularly, what we're interested in is protective antibodies. And then we leverage major advances in antibody isolation. So we can now sort B cells and come up with other ways of isolating antibodies from patients, and then functionally characterize them, identify those that are most potently neutralizing, broadly protective. We can down select and identify really what we would consider to be the top 5, 10, 20 best antibodies that humans can make against that pathogen. Hopefully these antibodies are also protective. And then the goal of the vaccine development is to create immunogens and immunization regimens that will re-elicit these antibodies in the vaccine recipients. And so we come in here with the structural biology and try to determine the high-resolution structures of the antibodies in complex with the antigen. That provides insight into the mechanism of antibody-mediated neutralization. It can also provide insight into the role of the viral glycoproteins that it's binding to. And then importantly, provides access to the chemistry at the interface. And this allows for structure-based and computational approaches to antigen design. So here we can see maybe this part of the antibody is binding this red portion of the antigen, not the purple portion. Maybe the purple portion is immunodominant, elicits non-neutralizing antibodies. So you could do things like computational epitope scaffolding and transplant that red region onto maybe a self-assembling nanoparticle and really try to focus an immune response onto that part. So a lot of this is really based on immunofocusing and eliciting best-in-class antibodies that were previously isolated. With the structural information, there's several different types of computational design approaches. You could use sequence-based design, where maybe you're trying to provide breadth against many different isolates. And so you could use, so we have like different isolates here, sequences 1 through 5. You could come up with ideas of using like consensus sequences to design your antigen, or perhaps common ancestors that would share residues from both of those isolates. You do conformational stabilization, which will be the focus of my talk. Some of these viral glycoproteins exist in multiple conformations, particularly pre-fusion and post-fusion. One of these is likely to have or be a better immunogen, and so we need to determine how to stabilize them and immunize with the correct conformation. And then other structure-based design approaches, like as mentioned, this epitope scaffolding, where maybe we can take an antibody binding site like this helix loop helix from this protein, graft it on to another scaffold, and then immunize with that to really focus the immune response onto just one portion of that particular antigen. And then subsequently, these can be delivered in different ways and even presented on self-assembling nanoparticles. We'll talk about, for the first part of the talk, our class 1 viral fusion proteins. These are all synthesized as single-chain inactive precursors. Here you see several of the most common ones: the coronavirus spike, HIV-1 envelope, influenza hemagglutinin, and then the F proteins from paramyxoviruses and pneumoviruses. These have generally evolved from one or two original common ancestors. They need to be proteolytically activated at a conserved site. And you can see that this has the two components. There's a membrane fusion component and then a fusion suppressive cap. And the membrane fusion machinery, that's been conserved and evolved from a common ancestor. It has the 4 elements needed to promote membrane fusion, which is a transmembrane domain that anchors into the viral membrane, a fusion peptide that gets released after the proteolytic activation that generates this little hydrophobic stretch of amino acids that will eventually harpoon into the target cell membrane, and then 2 heptad repeats, so stretches of 7 amino acids, heptads, that have hydrophobics at positions 1 and 4. Those will end up interacting with each other and forming a highly stable coiled coil and later a 6-helix bundle. And then you have, some of these have fusion suppressive caps. So we like to think of these as generally being spring-loaded. They want to undergo a conformational change from pre to post. And the fusion suppressive cap, like a mushroom, sits on top and prevents early activation. And generally, you couple like receptor binding, in the case of coronavirus, receptor binding to the S1 subunit or RBD, destabilizes it, causes shedding, and then you get refolding of S2. So this is what we like to think for how these would sit in a membrane. They initially fold into this metastable state. So it's not the lowest energy state. That's the post-fusion state, but they start off in this metastable pre-fusion state. There's some activation energy, a triggering event that causes it to refold. You then have the harpooning of the hydrophobic fusion peptide into the host cell membrane. And at this point, it adopts what we call a pre-hairpin intermediate, where the protein is spanning both the viral membrane and the host cell membrane. This then begins to collapse in half as the coiled coils walk toward each other. This brings the host cell membrane and viral membrane together. Eventually, the protein adopts the highly stable post-fusion conformation, And then the host membrane and viral membranes fuse, and you now have a fusion pore, so the genome of the virus can enter the cell. And at that point, the cell is infected. And what we want to do is generate antibodies and maybe small molecules that can bind to the initial pre-fusion state and prevent the conformational change from occurring. Right, so if we can either purify and isolate antibodies or use those for passive prophylaxis or generate these types of antibodies through vaccination, they can bind, prevent the conformational gymnastics, and then the virus cannot enter, and that provides protection. A problem is that the pre-fusion state is metastable, and so there's a nice paper from David Baker and David Agard describing this. And so metastability means that it's thermodynamically unstable but kinetically trapped. So it's not the lowest energy, free energy state. It's higher, but it's kinetically trapped until there's a triggering event that gets it over the hill. And so one of the problems has been when you try to work with these proteins and purify them, sometimes just even look at them, they just quickly transition to the post-fusion state. And so many times you're immunizing or working with the post-fusion state when the ideal antigen may be the pre-fusion conformation. And so we did a lot of work on this back when I was a postdoc here at the Vaccine Research Center in Peter Kwong's lab, and working with Barney Graham, was able to determine structures of the post-fusion and pre-fusion conformations of the respiratory syncytial virus F protein. These proteins are trimeric, so there's 3 copies of each F protein. And you can see that one protomer is in ribbons, one protomer is in gray surface, and one protomer is in white. We can then just look at one individual protomer, the one in ribbons, and we can start to appreciate the conformational changes that occur and the differences between the pre- and post-fusion state. So you see about half the molecule that's in white does not undergo a conformational change. So these amino acids are within about 5 angstroms. So you can see there's a helix loop helix. That's the same between pre and post. There's a large beta sheet. That's mostly the same. But then the N and C terminus of the F1 subunit undergo really substantial refolding and conformational rearrangement. So the C terminus of F1 pivots around this amino acid here, and this flips all the way around to form the outer helix of this highly stable 6-helix bundle. And then the N terminus of F1 completely refolds. You have the hydrophobic fusion peptide, which is sequestered inside the center of the protein. You then have a helix turn helix, this beta hairpin in yellow and green, and then the final alpha helix 4 in blue. All of this flips around and polymerizes the central helix and forms this highly elongated alpha helix in the post-fusion state. So you even have these elements like the beta hairpin in yellow and green are refolding and transitioning into an alpha helix. And so it's a really substantial refolding and conformational change. And so with this information, we're then able to identify stabilizing amino acid substitutions that would lock the protein in the pre-fusion state, so that way we could use it as an immunogen. And so to do this, we generally try to identify several classes of stabilizing substitutions. Disulfide bonds are always ideal when we can find them, particularly if we can get one cysteine in a region that moves and another cysteine in a region that doesn't move. So then they can form a covalent disulfide bond, staple those 2 regions together, and prevent the conformational change from occurring. We can also identify cavity-filling substitutions. Many times in these proteins, these have evolved to be somewhat unstable. So there's little pockets and cavities that the evolution has created instability. And so we can find those and fill them. So in this case, there's normally a smaller amino acid serine pointing at this cavity. And so we replaced it with a larger phenylalanine that now fills the cavity, stabilizes the protein, and makes it unfavorable to pull that phenylalanine out and expose it to solvent. Sometimes there's charge repulsion. In this case, in RSV, there's a lot of negative charge, 6 negatively charged amino acids. You can mutate these to asparagine or glutamine to reduce the negative charge, reduce the repulsion. Sometimes we engineer in salt bridges. So we tried to, normally this is an arginine, positively charged arginine next to a positively charged lysine. This was mutated to a glutamate by Johannes Langendijk's group for HIV on, and now you get a positive-negative salt bridge to help stabilize it. And then finally, prolines, which we love. And they work really well, and that's been shown for influenza, HIV, RSV, and then later for coronaviruses. Prolines that cap alpha helices, or in these regions that need to undergo a coil to helix transition, putting prolines in there which are rigid, relatively inflexible, help prevent the conformational change from occurring. We used to do this just by eye. Now there's a lot of servers, tools, AI/ML models to assist with this. And so it's kind of an exciting time for doing this type of design. Ultimately, back in 2013, we settled on a single disulfide bond that linked this moving region in blue to this moving region in gray. And so that formed this covalent linkage, and then added in these cavity-filling substitutions at the apex. And that was sufficient to keep the molecule in the pre-fusion state. And then when Barney and colleagues at the VRC immunized mice and rhesus macaques with a prime boost, and then looked at the RSV neutralizing antibody titers, we could see that higher is better. With the post-fusion form of the F protein, we were getting NUT titers in the hundreds. And then with the pre-fusion form of the F protein, we were getting titers in the high thousands, close to 10,000. So really a substantial increase using the pre-fusion conformation of the F protein as an antigen. Later, when I was a professor at Dartmouth, we worked with a company called Adimab and Laura Walker to try to provide an immunological basis for why using the pre-fusion protein as an immunogen elicited such higher neutralizing antibody titers. And so working with Laura, they were able to isolate 364 RSVF-directed antibodies from 3 adult donors. All of these were expressed and purified, and we characterized for affinity, their epitope, neutralization potency. And if we look at the most potently neutralizing antibodies, we can see that 60% of them target only 2 epitopes, or antigenic sites, that we call antigenic site 5 and antigenic site 0. And importantly, these are the pre-fusion specific antigenic sites. So those sites are only found on the pre-fusion form of the protein, and they're in a completely different conformation in post-fusion. They essentially don't exist in post. So if you immunize with post, you'll never be able to elicit all of these pre-fusion specific antibodies that are highly neutralizing. And so the extent to which pre-fusion stabilization works really depends on the percent of antibodies that people make that are targeting epitopes exclusively found on the pre-fusion conformation. So we had all that work done back in 2013 with the stabilization of the antigen. It went through the normal decade or so of clinical trials. And then in 2022, both GSK and Pfizer announced top-line phase 3 data from their RSV vaccine candidates in older adults. And then last year in May, the FDA approved the first RSV vaccine, Arexvy, from GSK, which is based exactly on the dsCAV1 molecule that we created. And so this is a picture Barney sent to me in the fall of last year, where he's getting vaccinated with a pre-fusion F molecule while holding a pre-fusion F molecule that he helped create. So I think that's really cool, and some of the power of the Vaccine Research Center and what can be done here. I then left the VRC in 2013 to start my own lab at Dartmouth that I mentioned. And Barney and I wanted to keep working together. We were trying to think what else could we apply structure-based vaccine design to with other viruses. And this was around the time that the Middle East respiratory syndrome coronavirus had emerged in 2012, 2013 in Saudi Arabia. And that was the second coronavirus that had spilled over. We had SARS in 2002, and then MERS in 2012. And we really felt like, wow, we should try to figure out how to make optimal coronavirus vaccines, anticipating there would be additional spillovers in the future. And the nice thing was that they're both spike protein from coronavirus and RSV-F protein. Again, they're both class 1 viral fusion proteins. We felt like we could use a similar paradigm where we first needed to obtain a 3-dimensional structure of the pre-fusion state of the coronavirus spike, identify stabilizing substitutions, and then test it as a vaccine immunogen. And so that's what we set out to do back then. And it took several years, and it was complicated. We were trying to work on MERS. We could not get the structure. Ultimately, we worked with Andrew Ward at the Scripps Research Institute, and one of the other human coronavirus called HKU1, that's a cause of the common cold, and Andrew's lab was able to get this 4-angstrom cryo-EM structure of the pre-fusion HKU1 spike. And so this provided a lot of insights into the biology and function of the spike protein. We saw that the receptor binding domains were mediating trimerization. We knew that the RBDs would have to undergo a conformational change to bind receptors, otherwise there'd be substantial clashes. But for whatever reason, in this base state, all the RBDs were in the down conformation. It identified 2 additional subdomains, these smaller domains, SD1 and SD2. And then most importantly, it provided the structure of the spring-loaded fusion machinery, which ends up actually looking a lot like the RSV-F protein. So since that time, my lab, Andrew's lab, David Wiesler's lab, Tongqing, and others have solved a lot of coronavirus spike structures bound to antibodies, bound to receptors. And now we can start to make a nice little movie of how these work. I think this is better than my animation in the beginning. So you have the fusion suppressive cap, the S1 domain, the fusion machinery in S2. We now know that the RBDs hinge into an up state. Those bind receptor. It destabilizes S1, which falls off. You get that refolding and polymerization of the alpha helix and harpooning of the hydrophobic fusion peptide into the target cell membrane. And then the pre-hairpin intermediate collapses as those heptad repeats and coiled coils start to form the 6-helix bundle. It zippers up and then brings the viral membrane and host cell membranes together. So these are really impressive little molecular machines that have evolved to fuse their membranes with our membranes and infect us. And we're constantly trying to stop that from happening. So our goal at this point was to, so back in like 2015 maybe, trying to stabilize the spike proteins. And so here you can see the comparison of pre-fusion S2 and post-fusion S2 for coronaviruses compared with pre-fusion and post-fusion RSVF. And there's a lot of similarity. They all have, they have a central helix in blue that does not undergo much change between pre and post. It's just smaller in RSVF. And then you have these elements in the first heptad repeat in green, yellow, orange, red, all those same colors. And all of that's going to polymerize and stack and form that really long alpha helix that then shoots the fusion peptide into the target cell membrane. So we tried to do some of the similar tricks with disulfide bond, cavity filling substitutions, but we were having issues because for RSV, we had the RSVF structure, and we were trying to make RSVF variants. Here we had the HKU1 spike structure, but we were trying to make variants of MERS. And those proteins are only like 35% sequence identity. And so things like cavity filling and disulfide bonds, you have to be more precise than that. So we went back to the idea of using prolines, which worked really well for RSVF from Hans Langendijk and his group at Janssen. They ended up putting a proline between the central helix of RSVF and the first heptad repeat element. And that really just depends on conservation of secondary structure. And when we look at that same region in coronavirus spikes, we say that it's actually highly conserved. So these are different beta coronaviruses, different lineages, HKU1, MERS, SARS. What you can see is that they all have 3 conserved amino acids, RLD, 2 non-conserved amino acids, a conserved glutamate, and then most of the central helix is conserved. So my postdoc, Nian Shuang, started performing proline scanning and just mutating or substituting each of these amino acids for proline in MERS. And then we looked at the amount of protein expression, generally using that as a proxy for improved stability. And so the wild-type MERS spike, using the wild-type sequence, we could barely produce any. It was just this very faint band. But a single proline substitution gave us almost a 20-fold boost in protein expression, which is pretty amazing because these are like 1,200 amino acid proteins that trimerize, they're metastable, they're glycosylated, and mutating a single amino acid to proline can give you that much of a boost. Several other prolines in this region also improved expression. Ultimately, we chose the one that had 2 prolines next to each other that we called 2P, and that was around a 50-fold boost. When we purify these spike proteins and look at them by size exclusion chromatography, see the wild-type MERS spike, there's just this tiny peak down here. The 2 proline spike, really nice peak, monodisperse. Wild-type MERS spike is a mixture of pre-fusion and post-fusion that over time irreversibly triggers and adopts the post-fusion conformation, whereas the 2 proline MERS was homogeneous pre-fusion spike. We then sent the protein to Barney's lab and Kizmekia, a postdoc in his lab at the time, and they immunized mice with a low dose, a 0.1 microgram dose, with either the 2 proline spike in red, the wild type in blue, or the S1 subunit in orange, and then looked at the neutralizing antibody titer, so higher is better. And again, what we saw is immunizing with the 2 proline form of spike elicited a 5 to 10-fold higher neutralizing antibody titer than wild type or S1. It wasn't as big of a difference as what we saw for RSV, but still a significant difference. What's also nice about this stabilization trick is that because the region is so well conserved, we could put the same 2 prolines in any beta coronavirus. So we did this for SARS from 2002, and we could see the wild type in red, 2 proline in blue. Wild type SARS is a mixture of pre-fusion and post-fusion. 2 proline SARS is all pre-fusion. We did this for HKU1, which the wild type itself we could already make enough of and was sufficient for the cryo-EM studies, but adding in the 2 prolines again gave us a nice boost. And we even cloned a bat coronavirus, WIV1, and showed that the 2 proline worked. And so we published all this back in 2017 and patented it. We were a little disappointed it got rejected from 5 journals. And so that was tough. Nobody found this to be significant, and coronavirus outbreaks are regionally contained, and some other comments that were interesting. But we persevered, and it was published in a nice journal, PNAS. And then I moved the lab to UT Austin in 2018 to have access to cryo-electron microscopes. And so then we were really well prepared when in late 2019 we became aware of these pneumonia clusters of unknown cause in Wuhan. By early January, Chinese researchers had identified the novel beta, the agent as a novel beta coronavirus. On January 10th, they released the sequence online. And within 10 days, Nianshuang had cloned multiple plasmids containing the 2 proline stabilized spike. 10 days after that, my grad student, Daniel Rapp, purified the spike protein and started cryo-EM data collection. And by around February 10th or 11th, we had submitted a manuscript to Science and BioArchive describing the structure of the proline-stabilized SARS-CoV-2 spike. So we went from a genome sequence online to a submitted manuscript in 1 month, and we're never going to do that again. But it resulted in a science paper. It came back with the reviews in 48 hours and no revisions, which was amazing. We were trending on Reddit, so that was my phone screen capture. The paper's been cited over 10,000 times, so it's all downhill from here for me. But it's good to have a good one. And, of course, what we're most excited about is that all 4 of the COVID-19 vaccines authorized for use in the United States use the pre-fusion stabilized spike protein. So Moderna's mRNA 1273 contains the 2P mutations at 986 and 987, as does Pfizer and BioNTech's Community. Johnson & Johnson, they had the adenovirus, Ad26 vector. They also used the 2 prolines, and as did Novavax. So again, another great collaboration between academia and government and working with Barney that's led to antigens being incorporated into highly successful vaccines. We then went on to make an even more stabilized version that we called Hexapro. It contained 4 additional prolines. That substantially boosts expression of 2P. So 2P we could make a little bit of, maybe around 10, no, probably a couple mgs per liter of 2P. And then Hexapro we were getting closer to 32 mgs per liter. We determined a cryo-EM structure. There wasn't much of a change between Hexapro and 2P. This work was all led by my postdoc, Qinglin Xie. Rather than sending this plasmid out to a bunch of labs, we just added it to Antigen, and they've now distributed it to 227 labs around the world. So a lot of researchers working on coronavirus are using the 6P spike for structural studies, antibody isolation, and it's been incorporated into several new vaccines for low and middle income countries that we're really excited about. And this is now approved for use in Thailand. That was partnered with PATH. So it's called HXP GPOVAC, and so that's the Hexapro. And in Mexico, it's called the Patria vaccine, and so that's being used as a booster as well. So since that time, we've continued to work on some class 1 viral fusion proteins, but we've really been trying to push structure-based vaccine antigen design to other viruses and even some bacteria. The viral fusion proteins, you have the class 1s that I mentioned. You have class 2s that generally, they're a little different. Sometimes they start off as dimers or tetramers or higher order. They then go through an intermediate and then end up as a trimeric post-fusion molecule, like flaviviruses and bunyaviruses use class 2, and then some class 3s like herpes viruses. We've published recently on class 3, so I was going to talk about some of the work on class 2. And one of the ones that one of the viruses we've been focusing on that uses the class 2 viral fusion proteins is Crimean Congo hemorrhagic fever virus. So this is the most widespread tick-borne viral infections in humans. It uses these hard-body ticks as the vector. Generally, the life cycle just involves the small mammals and birds. It can eventually get on livestock and ungulates. The infection with CCHFV in these animals just causes a transient viremia. The animals don't even get sick. But when humans get infected, you get a hemorrhagic fever with a case fatality rate of around 10 to 40%. And so we're not normally part of its life cycle. We're more of a dead-end host, but it's particularly bad for humans. And the endemic zones are expanding. There's been some cases in Europe, Spain, and Italy, in part due to increasing temperatures, migratory birds, and livestock trade. CCHFV belongs to the Neroviridae family. So here are the Bunia viruses. There are 5 families indicated with the red human that can infect humans. And so these are now a major focus of some of the initiatives funded by NIAID, like these revamp grants that we're a part of, to focus more and try to generate tools, reagents, antibodies, and vaccines for Bunia viruses. These are trisegmented viruses. Their genomes are trisegmented. They have small, medium, and large genome segments. The medium segment, or M segment, encodes the glycoprotein precursor complex. And so for hantaviruses, it has a relatively simple M segment. It has the GN protein and GC. And GC is the class 2 viral fusion protein, so that's going to fuse the viral membrane and host cell membrane. And GN is an accessory protein that helps keep GC in the pre-fusion state and is sometimes involved in receptor binding. But for CCHFE, their M segment's a lot more complicated. It still has GC, which is the conserved fusion protein. It also has this NSM, a smaller GN, a protein called GP38 that was thought to just be a secreted molecule, and then a mucin-like domain. And so we've been working as part of this large consortium I'll show you a picture of called Prometheus. Is funded by U19 to really try to understand and get more structures and antibodies and create antigens for CCHFV. One of the interesting things about Bunia viruses is that the GN-GC heterodimers oligomerize generally into higher order states. So we can see for the orthobunia viruses, these will end up forming these tripodal complexes. For hantaviruses, GN and GC associate to form tetramers, and then those tetramers form lattices. For phlebiviruses, you get pentamers and hexamers. One of the things we still don't know, unfortunately, is what the higher order oligomerization is for CCHFV. We're still trying to analyze that. It's a BSL-4 pathogen, so we can't just directly visualize it under the microscope. There was a group in 2018 that published using Hazara virus, which is related to CCHFV but only infects animals. But it was still difficult to tell if there was a higher order assembly. So that's still an unknown question. This is the Prometheus group. So it was a CEDAR, this U19, led by Kartik Chandran's group at Albert Einstein. And so we've been working together for 5 years now. And I think have really helped push the field forward and created a lot of interesting reagents and insights into the CCHFV. Some of the initial work that we were excited with, mostly with Kartik, MapBio, and again, Adimab, they were able to isolate 361 monoclonal antibodies from 4 CCHF convalescent Ugandan donors. These were all expressed and purified, characterized. The majority of them, more than 75% from each donor, were GC specific, whereas some were able to bind this protein called GP38, which is thought to be secreted. Laura's group performed some yeast-based competition binding studies, and they were able to identify around 6 antigenic sites, with antigenic site 1 being the most dominant. So 173 antibodies were targeting site 1 compared to smaller numbers for the other antigenic sites. The GC-directed antibodies against numerous sites were potently neutralizing, whereas all the GP38 antibodies were non-neutralizing. These were then tested at USAMRIID, Andrew Herbert's group, and the lead neutralizing antibodies and their combinations were able to afford prophylactic protection, but not therapeutic protection from challenge. And so these are mouse experiments. And you have to do these in like IFNAR knockout mice or using an antibody against the interferon receptor in order for CCHF to actually cause disease in the animals. You can see if these are given, if the antibodies were given 1 day before challenge, then these neutralizing antibodies were able to protect. But if they were given 1 day after, then there's very little protection that was afforded. But still, in terms of prophylaxis, these could still be interesting antibodies to maybe help protect people who are going into an outbreak of CCHFV or in an endemic zone. What was really exciting is John Lai's group, who's part of the consortium, created a bispecific antibody. And they took 2 of the best antibodies, one against site 3 and one against site 1, and they made this dual V-domain chimera. And interestingly, this bispecific was protective when given 1 day after challenge. And so we're very excited by the bispecific and are working up methods of producing it in scale. But we wanted to understand the mechanism behind how these antibodies were neutralizing and perhaps the basis of the synergy. And so Akash Mishra, a grad student in my group at the time, was able to crystallize the individual Fabs as well as a complex of the GC protein bound to each of those 2 site 1 and site 3 antibodies. And so in this case, we saw GC. We were just getting a monomeric form of GC. We also were put in touch with Felix Ray's group, who we heard had a post-fusion trimeric structure of GC. So we combined forces to make one nice manuscript to provide more insight into the GC protein. We can see the monomeric form. There's a domain missing domain 3 we couldn't observe, but in the post-fusion trimer, this flips around and helps stabilize the very low energy post-fusion state. If we look at the site 1 antibodies, which were most immunodominant, these recognize the fusion loops. So for the class 1s, they have a fusion peptide, which shoots into the membrane. For class 2s, they contain 2 or 3 loops that have hydrophobic amino acids like tryptophans and phenylalanine that will just insert into the outer leaflet of the membrane. What's interesting is that the fusion loops themselves can actually exist in a pre-fusion and post-fusion state. And so this was, we can look at the structures for Maporelvirus, which is a hantavirus that Felix Ray's group had determined structures of. And you can see that the GC fusion loops in the post-fusion state have all the hydrophobic residues sticking out, these tryptophans, phenylalanine, tyrosines, ready to insert in the membrane. But in their pre-fusion structure bound to GN, now the phenylalanine is in this down position, the tryptophan has moved down, and so the presence of GN helps reorder and repack the fusion loops and hide the hydrophobic residues. What we saw is that in their post-fusion trimeric structure, the fusion loops were, of course, in the post-fusion state with exposed tryptophans, but the antibody that was potently neutralizing is actually recognizing the post-fusion conformation of these fusion loops, suggesting that it's engaging the fusion loops after GN has dissociated, but prior to insertion of the fusion loops in the target cell membrane. Antibody 36121, which is against antigenic site 3, and most of the other antigenic sites that we've solved structures for now, these bind the monomer, but are incompatible with binding to the post-fusion trimer. And so if the antibody is bound, the other protomers cannot bind. There would just be substantial clashes. And so ultimately, we think that you get GN dissociation, GC refolding, insertion of the fusion loops into the target cell membrane, collapse of the pre-hairpin intermediate, and then adoption of the post-fusion state, and that these 2 antibodies are targeting 2 different steps of the fusion process. The site 1 antibodies combine the released fusion loops after they refold into the post-fusion conformation. This will prevent membrane insertion. And then the site 3 antibodies would prevent the assembly of the highly stable post-fusion conformation. And so you're targeting different steps, and perhaps that's why we're getting synergy. We've still been trying to do more work on assembling structures of the larger complex for CCHFV. Back in 2020, we also solved the structure of GP38, but that didn't provide a lot of insights. We were hoping it was going to look like something and provide function, but it did not. Again, it was thought to be secreted and is the target of protective but non-neutralizing antibodies. And so we're really trying to get structures of GN or a complex of all of these together and maybe the higher order oligomeric state. AlphaFold 2 coming out was actually a really big advance. And Felix Ray's group published a nice review on this where they used AlphaFold and showed that the structure of hantavirus GN would look very similar to the AlphaFold 2 model of CCHF GN plus GP38. And so maybe GP38 isn't just a secreted protein. It's actually sort of GN-like, but ultimately gets cut and dissociates. And so we wanted to test this model. So Elizabeth McFadden, a grad student in my lab, she used the AF2 model to identify and design a stabilizing disulfide bond that would link GN in orange to GP38 in blue. All right, so we made this construct, so we would express this protein. The mucin-like domains would get cut off so that that wouldn't be seen. And we would see a single band for GP38 and GN, which we could see there. If we added in this predicted disulfide bond, then we got a very large boost in protein expression, indicating that the disulfide is forming and that the model, at least in that region, is likely to be correct. We were then engineered in a furin site that would allow cleavage between GP38 and GN. So on a non-reducing gel, we could see 2 bands for GP38 and GN. And then on a non-reducing gel, where the disulfide would form, that shifted up to a single band, again showing nicely that the disulfide bond that we designed is forming. And without the disulfide bond and allowing cleavage to occur, we actually get no expression. So it's very unstable, just GP38 and GN without any of the without the stabilizing disulfide bond. Interestingly, the protein is actually quite stable. The GP38 GN with a disulfide bond had a melting temp of around 60 degrees. Elizabeth was able to crystallize and solve the structure of GP38 GN, and then we wanted to try to get the full complex with GC. So we took this approach that Felix Ray's group used for hantavirus, where they link GN and GC with a glycine linker that could be cleaved. And we took our construct with the engineered disulfide bond and then added a glycine linker with GC. This actually expressed very well. Had a melting temp of around 65 degrees, and it allowed Elizabeth to obtain a cryo-EM structure to 3.4 angstroms, showing the complex of GN, GC, GP38. Importantly, some of the antibodies we had isolated previously were used here to add some mass and help improve signal and contrast so we could get the structure. What it shows is that GN is really wedged in between GP38 and GC, and that overall the GP38 GN for CCHF looks like hantavirus GN. Interestingly, whereas in hantaviruses, GN uses this proteinaceous loop to help cover the fusion loops from GC. Here, it's actually an N-link glycan that's emanating out from GN that interacts with and stabilizes the hydrophobic fusion residues of GC, which we thought was really interesting. And then Elizabeth made a movie to show the transition of the fusion loop dynamics between the pre-fusion and post-fusion state. So you can start to appreciate how even just these loops can still undergo conformational change and both hide and then expose the fusion residues, or the hydrophobic residues, for insertion into the target cell membrane. We then worked with Usamaryd and Stephanie Monticelli's group to immunize with these proteins and see how they worked. So here we're using these C57 mice with an antibody against the interferon receptor. There was a prime boost followed by a challenge. And what we saw was that all the proteins with GP38 provided some partial protection, like 40%. GC alone, despite eliciting neutralizing antibodies, does not provide a lot of protection. When we look at it a little bit more closely, and we look at those groups and their ability to elicit GP38 binding antibodies, our engineered construct in pink, the heterotrimer that we got the cryo-EM structure of, that actually worked really well in eliciting GP38 antibodies. And all the mice, 4 out of 5 mice that elicited strong GP38 antibody responses survived, whereas 4 of them didn't really elicit anything. So we actually think this immunogen is doing something. And if we can improve and get a better response from all the mice, we might be able to get up to 80 or 90% protection. So to summarize the work on CCHF, this big consortium that's ending, although will be reincarnated under a new U19 called Revamp. We've been able to isolate GC and GP38 antibodies. No GN antibodies isolated to date, but we think that's in part because GN is actually really quite small relative to, it's kind of wedged between GP38 and GC. We've determined structures of GP38, GC, the heterotrimer. We think that this suggests that GP38 is not just a secreted protein. It actually is a structural protein initially, and then after proteolysis, GP38 dissociates from GN, and then GP38 may have an additional role. Working with Eva Harris's group, she showed that it is similar to dengue NS1 and produces some vascular leak, and maybe that's why antibodies targeting GP38 are non-neutralizing but protective. We're continuing to engineer the heterotrimer as a vaccine antigen, different ways of displaying it. We're still trying to characterize the higher order oligomerization state for CCHF glycoproteins. And then importantly, we now have these antigens as new probes to go back and try to isolate antibodies that maybe bridge across GC, GN, and GP38 that could be useful prophylactically. Additionally, we're doing a lot of work with funding from CEPI, as I mentioned, the NIH revamps, a new ARPA-H grant, and really trying to develop these antigen libraries for disease X. ARPA-H is more for herpes viruses. CEPI paying us to create AIML-based tools for rapidly designing stabilizing substitutions. So we're sort of going through the 25 viral families known to infect humans and creating these antigens, testing them, and having them ready, much like what we did with MERS and having the 2P ready before SARS-CoV-2. We want to identify similar stabilizing substitutions that could be used when the next virus spills over. Working with Jimmy Gallagher, friend and colleague at Houston Methodist, we've made stabilizing proteins for all of these different viruses and viral antigens. And so in less than 1 year, we made 8 viral glycoproteins, and over 3,200 variants have been assessed. And we have many more in the queue now. So I've highlighted a lot of people in my lab. Again, this has been really collaborative with great collaboration with Barney Graham and colleagues at the Vaccine Research Center, Andrew Ward's group for the initial work on cryo-EM of HKU1, all the Prometheus colleagues in the CCHF group, generous NIH funding, both R01s and U19s. Thanks very much for your attention, and I'm happy to take any questions.
Thank you. Tour de force indeed. Any questions from the audience? Nothing has come in online yet.
That may have been more structured than that's okay.
Just don't want to force anybody.
Hello, great talk. So I have a question for your antibodies that target post-fusion.
Only post-fusion or also bind post?
Also post-fusion.
Okay.
Where do you think that's happening? Is it happening outside the cell when the cell is infected, endosome, receptor binding? When are those epitopes exposed?
That's a good question. So the antibodies that also bind post, they bind pre as well. And so that's why many of those are neutralizing, is because of their activity to bind pre. And so we think that those are just binding pre-fusion on the surface of the variants. Some of the other ones may be more interesting if they're binding like epitopes that become transiently exposed. And in some cases, there are for some viruses where maybe you need to be taken up into the endosome, and then cleavage occurs, and now that exposes the site. So in that case, you need the antibody to get potentially taken up into the endosome, thinking of like filovirus Ebola as an example. And so Kartik's group has done some engineering with bispecifics. That way, like one arm binds and the other arm is ready once it's taken up in the endosome, which is kind of interesting. So I think a lot of cool antibody engineering can be applied here.
Yeah, really good talk.
Thanks.
And you showed in the CCHF virus, the AlphaFold predict one state, which is close to the real structure. Yeah. So I wonder, because you always have pre-fusion and post-fusion, can AlphaFold predict both in unknown viruses? Yeah,
it's a great question. It depends a little bit. People have created some models where you can use templates to help bias it. Yeah. But then you're kind of biasing it. Yeah. So that's on its own. It will. It may pick a pre or a post, or we've seen it can provide like a chimera, where some of the secondary structures more in pre, and others have flipped out and are more in post. Yeah, that has been a limitation, but that will probably improve as more structures are available, and the templates do help bias it in that direction. The goal for CEPI is really to maybe go from a novel virus spilling over, the sequence being isolated, AlphaFold structures of all the different states, AIML-based substitution identification, and then mRNA manufacturing without ever needing to go into a wet lab. Future. Yeah.
Hi, it's a very beautiful talk. You have shown that the membranes of viruses versus the membranes of the cellular membranes, and even in the cell, the endoplasmic reticulum membrane versus, they have different thicknesses.
Well, the viral membranes come from the cell membranes.
So
right? They're pulled, that's where the viruses get their membranes from.
So
yeah.
And the peptide embedded into these membranes has a length somewhat about 16, 17 amino acids, something like that. But if you have go into the other membrane like endoplasmatic reticulum, there's a smaller length. If you extend that length, the protein will go to the cell surface membrane. So that is important to be taken into consideration. What is the membrane length, or the thickness of the membrane?
Yeah.
I
would say most or all of these structures I presented have been from soluble ectodomains of purified proteins. We're now doing more structures on the surface of vesicles or viruses themselves, and we're actually starting to be able to see the TMs and maybe even some bound lipids and things into the protein. So we're excited to move away from purified proteins into more of their native context.
Okay. Question from the south side here.
So it makes perfect sense that blocking the prefusion domain from interacting with the receptor on the infected cell prevents infection. But what actually happens to the virus that is coated with the anti-prefusion antibodies?
Yeah.
Does that get phagocytosed, represented as cross-presented antigens as well?
Yeah, we know they can get taken up by macrophages. It could also be endocytosed and destroyed. There may be other outcomes.
Do they cause renal problems?
That I do not know.
I do have a question online. I noticed that your group didn't try to tackle the HIV-1 ENV protein. Is that envelope?
Yeah.
I don't know.
Yeah.
Which is also a class 1 fusion peptide. Is there a reason for that?
Because there's already great investigators working on it here at the Vaccine Research Center and elsewhere. And so we try to maybe go to some fields that people aren't working on or are a little understudied rather than competing with some of the heavyweights for HIV and universal flu and some of my colleagues here.
Okay, very well.
It's a diplomatic answer.
Dr. Shore?
Yeah, a wonderful talk. I'm just wondering, so you've shown lots of data derived from sequential experiments with different configurations of the various proteins. Can you now use that information that you've gained to go back and a priori look at a given protein and predict which configuration is going to be most immunogenic in a given virus? I mean, or do you, each time you work with a virus, do you have to go through the same sequence of experiments?
Yeah, I think it still has to be done empirically. I think the immune system is too much of a black box. I know people are trying to come up with AI-based immune systems and things, but and then even different animals and their repertoires could be different. So I think you really have to test it out. Ideally, we'd like to use antibodies as the guide, right? And so that worked very well with RSV in particular. Groups had already isolated pre-fusion specific antibodies. We knew they were super potent, and so the goal was really just trying to elicit them. For some of these other pathogens, it's hard to even generate antibodies from or get convalescent donor. So we really have to start over, I think, test empirically. And then sometimes you don't know. I mean, maybe one form tested as a protein subunit could give a different result as tested as an mRNA delivered. So I think you need to test it with different modalities, delivery, different adjuvants, and really tease it apart. We always think that regardless of what the optimal antigen is, it's probably not what nature evolved, right? So nature's evolving it to function and undergo these changes, not evolving it to be an optimal antigen. So we think we should always be able to do better than nature. We should always be able to bring antigen engineering and provide some benefit. And it may not always be but we need a lot of immunology and virology, and that's why the work is so collaborative.
Thank you.
Thanks.
Hi, Jason. So fusion peptide is so important, but based on all your structural analysis, and it's also quite conserved. So why haven't we seen the fusion peptide neutralizing antibody in all kinds of, say, class 1 viruses? Shouldn't we see more often of this type of antibody?
Yeah, it's a good question. I think part of it depends perhaps on the location of the fusion peptide in the pre-fusion state. So for HIV envelope, as you know, it's more on the lateral side, somewhat exposed. That's true for coronaviruses, paramyxoviruses. For RSVF, it's buried inside, and so it's really only transiently exposed. Right? So the B cells would have a very small window between after the virus has attached and then you have triggering. So I think for RSV, you just may not see a lot of them. It's also possible they exist and we just haven't found them, right? Many times you find what you go looking for, and a lot of it's bait-based discovery. And so I'm unaware of people that have tried to use fusion peptide as bait to isolate RSV F-directed antibodies, but maybe people have. But yeah, a lot of it's bait-derived antibody isolation.
Yeah. It should be accessible by fusion, so it should be accessible.
It should be accessible, but I think in some cases, like in the case of RSVF, the virus is already attached, and then you would still need to get some B cell in there to try to recognize. So I think there's both sterics as well as temporal things to consider. Thanks.
Hey, Jason.
Hey, Chris.
So you showed this very nice pipeline that you usually start with antibodies derived from patients.
If possible.
If possible. So what are other means of having a good starting material? Do you also start by immunizing, for example, llamas or alpacas or other sources of antibodies, like rabbits?
Yeah, so that's a great way. I mean, we've done it antibody independent as well, which is really painstaking because we can't really assess the conformation of any of the variants, and we ultimately need to look by EM, which is very slow and tedious. If you don't have the human-derived antibodies, then you should at least be able to generate reagents by immunization of small animals, and maybe best is to do some sort of mRNA-based immunization of the full wild-type protein, which we'd assume initially folds into the pre-fusion conformation and is relatively stable. You could immunize with post-fusion as well. Having antibodies, ideally you want an antibody that recognizes all forms and then an antibody that recognizes only pre-fusion. You could also have an antibody that only recognizes post-fusion, and the ratio then gives you the pre-fusion percentage. So people have immunized with just peptides that form the 6-helix bundle, which is only on pre. So sometimes we actually have to start off, and for some of these revamps, we do start off with a reagent generation and try to get those just from animals.
And do you have an estimate if you get better neutralizing antibodies from patients compared to other sources?
That's a good question. I feel like many of the best ones in the different fields are usually from humans. Yeah, I have to think if there's a case where a murine antibody was really good compared to a human. And again, but some of that's just bias. Maybe people haven't looked as hard in the one animal versus the other. Yeah.
Yeah.
Thank
Thank
you.
you.
Question. Thanks.
Okay, maybe one last question from online. You're holding up? Okay.
Yeah, I'll make it.
Beautiful talk. Thank you very much. Regarding the studies looking at GC stabilization via the end-linked glycan from GN, could the difference between the glycosylation machinery of ticks and humans, or whatever cell model was used to probe this interaction, produce differing results compared to what was observed?
Yeah, I don't think so. I think most of what the glycosylation that we're seeing laying onto the GN or the GC fusion loops is really just the base glycosylation. It was 2 glucanacs and the beta mannose. That's generally conserved in in all eukaryotes. And so sometimes you get differences on the branching or the terminal residues, depending on processing. But there is really like the more conserved core glycans that were responsible. So I don't think that would have as much of an impact.
Okay. I think we have no more questions. So first, I'll remind everyone there's a reception right outside. Everyone here, welcome to go. The CME number, code 57666, 57666. And lastly, let's thank our speaker.